you for that excellent summary, Dr. Zigbogel. We will hold questions until the end of all three speakers. The next speaker is Dr. Jennifer Guerriero from Dana-Farber Cancer Institute. Her talk is titled Macrophage and Microbiome Diversity in the Breast Cancer Microenvironment. Thank you.
Thanks so much, Justin. Hello, good afternoon. I'm delighted to be here to present on macrophages and microbiome in breast cancer. A couple of relevant disclosures before I begin. So just the goal today is to give a comprehensive overview of macrophages and microbiome in the breast tumor tissue, discuss the complexity, diversity, ontogeny of macrophages, discuss the clinical relevance, and then the therapeutic targeting. And then at the end, I'll try to link back to some of the work that Dr. Zigbogel just mentioned in the microbiome, as well as some of the other work that I'll present to try to connect macrophages and microbiome, specifically in breast cancer. So just a quick Immunology 101 on innate and adaptive immune responses. So innate immunity is composed of mononuclear as well as granulocytes. These cells respond non-specifically but quickly to infection, whereas adaptive immune cells are your lymphocytes. These are your B cells and T cells. They respond more slowly because they're responding to specific antigen and epitopes. So shown here graphically on the x-axis is time of infection. Innate immunity responds quickly. It's your first line of defense. And then T cells take a bit longer to activate, but they have the advantage of being able to facilitate memory immune responses. So upon second exposure, they can respond quickly to infection. As we know, T cells have been the main focus of cancer immunotherapy, and we now know that targeting T cells to reactivate them in the context of cancer has been tremendously successful for some cancer types, but not all. And this has really created opportunity to think about what other immune cells can be targeted for anti-cancer therapy. So indeed, there are a number of different types of immune cells within breast tumors. This is some really nice work from Brian Ruffel when he was in Lisa Cousin's lab. They looked at both normal as well as tumor tissue, both in the treatment naive and in the treatment setting. And they showed using all of these different subset markers that breast tumors are indeed composed largely of myeloid cells and T cells. Other work from Nelly Poliak at Dana-Farber has shown that both normal breast tissue, shown here on the left, as well as invasive disease, HER2 negative, HER2 positive, triple negative, are also infiltrated with myeloid cells and T cells as the two major populations, again broken up into different subsets. And importantly, when you look at the percent of leukocytes, CD45 positive cells out of total cells, the tumor tissue has a higher abundance of immune cells compared to the normal tissue. And if you look at this, the y-axis, it's up to 80% of the cells within the tumor are immune cells. So I think that's pretty striking. Now, this is CyberSort data from Dr. Pollard and his colleagues, in which they analyzed different types of cancer to look at the average immune cell composition, estimated fractions of leukocyte RNA shown here. And interestingly, macrophages represent the major population of immune cells in most human cancers, with breast being right here. And so I think this is visually quite striking, as demonstrated here. On the left is normal breast tissue, on the right is breast cancer. Immunohistochemistry was performed using the CD68 human macrophage marker, and you can see that the dark brown staining in the tumor tissue indicates the abundance of macrophages in the cancer tissue, but not the normal tissue. And importantly, we know that an increased macrophage density is associated with poor prognosis. Patients who are stratified by high density of macrophages in green versus low density in blue show that patients with more macrophages have worse relapse-free survival as well as overall survival. So why is this? Well, we know that monocytes circulating in the periphery are recruited to the tumor through different tumor-derived factors such as CSF1 or CCL2. Once the monocyte gets to the tissue, it matures into a macrophage. It senses that this tissue has low oxygen and poor nutrients, so really at no fault of its own becomes the type of macrophage that repairs wounds, and in doing so secretes factors that help the tumor cells grow and survive. These types of macrophages secrete factors that lead to angiogenesis. They secrete factors that break down basement membrane walls, leading to metastasis. They inhibit antigen presentation. they recruit suppressive T regulatory cells, and importantly, these types of macrophages inhibit CD8 positive T cell responses and function. And so, in general, we think that macrophages, or we know macrophages in tumors have a suppressive phenotype, and they are similar to the types of macrophages that repair tissue damage. On the other end of the macrophage polarization spectrum, we know that macrophages can also have anti-tumor function. They can identify and destroy malignant cells. These are the same types of macrophages that protect your body from bacteria or pathogens. So I think that this dichotomy of M1 and M2 is sort of getting phased out, and we encourage the use of sort of cell markers to characterize macrophages moving forward, and I'll get to that in a couple of slides. So because of the role of macrophages in the tumor, there's been substantial efforts to target macrophages for anti-cancer therapy. We can think of it maybe in two different strategies, either to deplete or to inhibit their pro-tumor function, or alternatively to reprogram them, push them from a pro-tumor to an anti-tumor phenotype. And certainly there's a number of compounds that have entered the clinic, some of which have been approved, such as the CSF1R agent that we'll come back to in a little bit. But there's a number of other targets being evaluated preclinically, and despite that I've spent my entire career studying tumor-associated macrophages, I would be the first to admit that we really need to learn more about these macrophages in the context of cancer so that we can more successfully target them. I don't think we really know how these macrophage modulating agents work. I don't think we know what subsets of macrophages they're working on, and we definitely don't know who's going to best respond to them. So why is that? Macrophages are really complicated. They're complex cells. So to start with, they have a diverse ontogeny. Generally, we think about macrophages as those that derive from the hematopoietic stem cells, from the bone marrow, to myeloid progenitor cells, to monocytes, and then when they reach the tissue, become macrophages. And indeed, tumors are full of bone marrow-derived macrophages, but each tissue in your body is 5 to 20% tissue-resident macrophages. These are cells that are derived from the embryonic yolk sac prior to hematopoiesis. Tissue-resident macrophages are very different than bone marrow-derived macrophages. They're self-renewing, they're long-lived, they have fundamental roles in tissue remodeling and then homeostasis. Again, very different than bone marrow-derived macrophages, which are short-lived and participate in wound repair. And so if you look at the contribution of tissue-resident macrophages, it's very different depending on the tissue type. So for example, in the brain shown here, there's early contribution of tissue-resident macrophages during embryogenesis, and these maintain throughout adulthood. This is in contrast to down here, the heart, pancreas, and breast that have early contributions of the embryonic-derived tissue-resident macrophages, but those are replaced throughout adulthood with bone marrow-derived macrophages. And there's beautiful work coming out of groups indicating that tissue-resident macrophages and bone marrow-derived macrophages differentially promote tumorigenesis in different tissue types. And so to continue on that point, the tissue-resident macrophages have very distinct tissue specificity, which means that the tumor-associated macrophages have important tissue specificity. So that makes sense, right? Because macrophages have important homeostatic function in defense, in homeostasis, in tissue remodeling. And so every type of tissue in your body has specific macrophages that are important for homeostatic functions. This is some really nice work from Ido Amit's lab in which they isolated tissue-resident macrophages from different organs. They performed a variety of assays. I'm just going to really talk about the RNA sequencing that they did, but they also looked at epigenetic changes between the different tissue-resident macrophages between tissues as well. But shown here is the RNA-seq analysis in which you can see that there's unique signatures between, or distinct signatures between tissue-resident macrophages from each of these organs, and again, distinct from the monocytes. And they could find distinct or unique gene signatures for each of these tissue-resident macrophages. And so I bring this up because I think that targeting tumor-associated macrophages is going to be different between different cancer types or tissue types. Now, I already alluded to this, but there's been inappropriate phenotype analysis to date. Tumor-associated macrophages are not M1 or M2. M1 or M2 actually has a definition. M1 macrophages are macrophages in a cell culture dish treated with interferon gamma and LPS. M2 macrophages are treated with IL-4 and IL-10. So you can imagine that this doesn't occur in nature. And to just hit that point home, this is a paper where murine macrophages were isolated from glioblastomas, and RNA sequencing was performed, and then that data was compared to published literature data sets in which M1 and different subsets of these M2 macrophages have been characterized at a gene expression level. And really, interestingly, only 60% of the genes in the tumor-associated macrophages isolated from these glioblastomas were expressed in any of these gene sets. Again, as proof of principle that tumor-associated macrophages don't cleanly fall into the M1 or M2 designations. And so, additionally, subset diversity, there's actually been a huge effort recently to better understand macrophages, their subsets across different cancer types. And so this is some work I had done maybe 10 years ago. It's multiplex immunofluorescence. We really just took 5 commonly used macrophage markers and stained a breast tumor, and then looked at the overlap between tissues. And Sandra Santagata, who's a pathologist I work with at Brigham and Women's Hospital, he said this is harrowing in terms of how underappreciated the TAM subsets are, where really there was very little overlap between these markers. More sophisticated work has been done by the Bowden-Miller lab, in which they used mass cytometry to look at 144 different human breast cancers. And among the really beautiful work that they showed, they pulled out 19 distinct myeloid populations that could be broadly broken into 5 categories. These include monocytes, the early immigrant macrophages, as defined by intermediate MHC class II expression, tissue-resident macrophages, tumor-associated macrophages, and then a more suppressive immature myeloid subset. And importantly, they concluded that the breast tumors were indeed enriched for immunosuppressive macrophage phenotypes, and that there was a high frequency between PD-L1 positive macrophages and exhausted T cells. So additional work has recently come out of the U Lab and published last year. This is sort of a lot of text, so I apologize for that, but I underlined sort of these unique subsets of macrophages that are coming out. This is an example from colon cancer, but we're now starting, and I think that this paper is really nice example of what we need to do. So what they did is single-cell RNA sequencing of colorectal cancer. They pulled out the myeloid subsets, and then they looked to see what they were doing. So for example, this PTLP positive tissue-resident macrophages, they were associated with blood vessels. They played a role in restraining inflammation and fibrosis based on a previous paper. The C1QA positive tumor-associated macrophages had high expression of genes related to phagocytosis and antigen presentation. SPP1 positive TAMs seem to be involved with angiogenesis. And so when they used anti-CSF1R therapy, which is a strategy that eliminates macrophages expressing CSF1R that are thought to be highly suppressive, they found that they could reduce the C1QC positive TAMs, but not this other subset of SPP1 TAMs. And interestingly, when you look at human data, patients who have high SPP1 and low C1QC TAM signatures correlates with poor prognosis. So to make that long story a little bit shorter, thinking about which subsets of macrophages need to be targeted for anti-cancer therapy may help us targeting the right subset of macrophages for cancer therapy or enhanced immunotherapy. And this is another paper that sort of confirms some of those findings. This is from Dr. Zhang and colleagues. And this was a really interesting dive into published data sets, as well as generating some new data sets on 15 different cancer types. And then they just analyzed the myeloid subsets. And it really showed diversity among myeloid subsets between cancer types. And so again, to just hit that point home again, that targeting macrophages will likely be different between different cancer types. So another complexity, location. I think we think a lot about where cells are within the tumor microenvironment and their relationship to each other, but this is some immunofluorescence we had done. The T cells are purple, the tumor cells are white, and CD68 and CD163 macrophages are in green. You can see the macrophages and T cells are right next to each other. So clearly there's some sort of interaction. They're talking to each other. And so I think we need to think about the spatial organization of macrophages as well. And so we've done some of this work ourselves. We've looked at BRCA wild-type TNBC as well as BRCA-associated TNBC. And really interesting, we've seen really different TMEs just stratifying by the BRCA status. On the right here, you can see there's just more color. There's more immune cells. We quantitated different T cell subsets as well as different macrophage subsets. The wild-type tumors are shown here on the left, and then the BRCA-associated is the bar on the right. Basically, in every category, there were more immune cells in the BRCA-associated tumors. Now, this is very much in line with some work that's led by Dr. Jeffrey Shapiro at Dana-Farber Cancer Institute, in which we are trying to understand PARP inhibitor resistance. And in doing so, we and others have shown that BRCA-associated TNBC, which is considered homologous recombination repair deficient, has high levels of cytosolic DNA. This high level of cytosolic DNA activates the SABCS pathway, leading to activation of type 1 secretion of type 1 interferons, such as CXCL10, CXCL9, CCL5, which recruits in immune cells. And so the hypothesis that this tumor-intrinsic mutation may regulate the TME, I think is very interesting. And so we we were able to demonstrate that in the previous slide I showed you, where those BRCA-associated tumors have more immune cells. Now, in work with, that again, Dr. Shapiro led, when you treat these tumors with PARP inhibitors, which are effectively used to treat BRCA-associated disease, you have further activation of the SABCS pathway, further infiltration of CD8 T cells, and PARP inhibitor efficacy was shown to be dependent on the SABCS pathway as well as the recruitment of CD8 positive T cells. And this was more proficient in BRCA deficient TNBC compared to proficient. And now, this work, as well as the work of others, set the stage to think about combining PARP inhibitors with checkpoint blockade based on the two different mechanisms of activating T cells. And so several trials have now launched looking at PARP plus immunotherapy. I'm starting by showing the Olympiad trial here on the left, which has been shown several times already at San Antonio. This was the study that resulted in FDA approval of olaparib, had objective response rate of 60%. This is Mediola, which combined olaparib plus anti-PD-L1, and you can see the objective response rate is about the same. These trials can't be compared head-to-head, but it does leave the question as to why the T-cell response is not more substantial and why the combination didn't lead to better improvement over single-agent therapy. And so the hypothesis our hypothesis was that macrophages may limit the therapeutic response to PARP inhibitors plus immunotherapy. So to test this, we use a mouse model. It's a BRCA P53 deficient model of TNBC. It was first described by Jos Jonkers and colleagues. We treat the mice for 5 days. We isolate the tumors and perform flow cytometry. You can see that there's a doubling of immune cells shown here by CD45. This is largely due to the increase in CD11b positive, F480 positive macrophages. The immunohistochemistry for CD11b positive myeloid cells is shown here, with the vehicle-treated tumors here and the substantial increase in staining of myeloid cells shown here. And importantly, we see a major increase in CSF1R as well as PD-L1 on this F480 positive myeloid subset. Because this data was so striking for the changes of macrophage phenotype, we asked if this was a direct effect of the PARP inhibitor on macrophages. So we turned to a human monocyte assay where we isolated CD14 positive monocytes. We differentiated the cells plus or minus olaparib and then collected the cells for analysis. And we see the same thing. There's an increase in PD-L1, an increase in CSF1R. This phenotype was also characterized by a change in metabolic reprogramming. They became dependent on lipogenesis. Importantly, these macrophages, when we put them with T cells, they not only inhibited T cell function, but they actually induced T cell killing. So we do think that this is proof of principle that macrophages are a liability for PARP inhibitor therapy. And so we tested the combination. We thought this was a rational approach. These macrophages increase expression of CSF1R after PARP inhibitor therapy, so we combined with anti-CSF1R therapy. This is a survival graph. Days of treatment are on the x-axis and percent survivals on the y-axis. You can see that the vehicle and CSF1R single agent had relatively no effect. The PARP inhibitor induces a median overall survival of 92 days. And when you add CSF1R to it, you extend the survival to 190 days. That's 100 100-day advantage in survival. These are mice. So we think this is really exciting data. Importantly, this was CD8 T cell dependent, and so we think this is proof of principle that both innate and adaptive anti-tumor activity is necessary for durable clinical outcomes. Now, CSF1R has been a topic of many pharmaceutical companies have developed CSF1R agents. They've not been a home run in the clinic. I've mentioned that before, but I think it's probably because it's not been the right setting. So this is work from the Roche team led by Dr. Rice with their CSF1R agent. So diffuse type giant cell tumors, they're characterized by a CSF1 translocation. So by definition, they have a lot of CSF1, which means they have a lot of CSF1R positive macrophages, as shown here in the immunohistochemistry. When you target CSF1R macrophages with this agent, these are largely depleted. And in this clinical trial, 5 out of the 7 patients achieved partial responses, and there were clinical objective responses in this particular type of malignancy. Now, another compound, anti-CSF1R agent, this Plexicon compound, was recently FDA approved for this type of disease. And so I use the PARP inhibitor as another example of a specific, another example of how a rational approach using CSF1R might be potentially successful. And so, in terms of the complexities, I've told you that the spatial, I've added to this list by showing you spatial organization, that this BRCA tumor intrinsic mutations may regulate the tumor microenvironment, that we should consider responses to therapy. The PARP inhibitor made these macrophages totally different. They have a different transcriptional profile. They express different proteins, so that needs to be considered. So what about the microbiota? So building on what Dr. Vogel just presented, the microbiota of the gut and the breast tissue is really fascinating and really important. So this is a slide courtesy of Dr. Jennifer Wargo, where she shows that 99% of the human body is microbial. I think that's just fascinating. And so there's been substantial advances in next generation sequencing. So now we are able to better understand the role of the microbiome, excuse me. And so, indeed, the breast cancer microbiome, so now we're talking specifically about the breast tissue, not the gut, and there's great diversity among species. This is, on the x-axis are different cancer types, on the y-axis is bacterial content, and so 60% of breast tumors are positive for bacterial DNA. That's the highest among these cancers that were tested. There's been reported differences between the microbiome of benign versus malignant tissue, and specifically and Dr. Vogel just mentioned that, abundance of bacteria that are associated with double-strand DNA break of cancer cells, so some maybe causal effects here. In addition, there's been correlations between breast cancer subtypes, ethnicities, responses to chemo and immunotherapy, as well as toxicity. So why is this important? So I alluded to it a little bit with the DNA break. So the tumor bacteria and their metabolites can manipulate different signaling pathways, so they should be considered, not just on the tumor cells, but also on the immune cells. We know that the the microbes may activate innate immunity, such as macrophages, through toll-like receptors. And some evidence from colorectal cancer has shown that that different species can actually cause suppression of immature myeloid cells, such as the MDSC population, thereby further suppressing T cell responses. So we know that breast cancer has rich and diverse microbiome. So shown here is a diversity index of the microbiome. Again, breast cancer has the highest diversity of the microbiome, and it has the highest. This graph here shows the number of species per sample. So also has the highest number of species compared to all these other breast cancer types. Something that I thought was particularly interesting was just the presence of LPS and LTA within the tumor, within the breast tumors. And I think a lot of times we think about the breast as a sterile environment. It's really not. There's a lot of exposure to external factors, such as women nursing babies and other factors. So this was a really this is some sophisticated work from Dr. Robertson and colleagues, in which they used an analysis called a pathochip, and so it's a parallel DNA and RNA detection of different microbial signatures. And so shown here are viral signatures, bacterial signatures, fungal and parasitic signatures of these different triple positive, triple negative, HER2 positive, or hormone receptor positive breast cancer. And what you can see is that some species actually are common among the different subsets of breast cancer, but some are unique to different subtypes of breast cancer. Interestingly here, the normal tissue has no, there is no viral signatures found in the normal tissue here. Similarly for bacterial signatures, there were some common signatures as well as, some common signatures as well as some unique signatures. And then the fungal and parasitic signatures actually had no common signatures among subtypes. So I think very, fascinating in terms of the subset diversity. Again, which species might be associated with causal malignant factors that contributing to malignancy should be further examined, but there's other factors that have been reported that are known oncogenic viruses, and others that have been reported to be responsible for angiogenesis. So in terms of outcome, specific microbial signatures may correlate with outcome, especially in the case of triple-negative breast cancer. So on the x-axis here are different signatures derived from the previous slide I just showed you, and then patient outcome is either alive in blue or deceased in red. And you can see that there are signatures that really do correlate with outcome. Looking specifically at the different species, there are species that correlate both with better and poor responses. So I have just one more slide. I think that, we should think very carefully about both the gut and the tissue microbiome. There's been some beautiful work by Dr. Wargo and Goldman-Schmidt and Lee in this area. So this is a little bit complicated, but basically what it's showing is the species of the different microbe components shown here, and then responses to CTLA-4 and PD-1. Blue indicates enriched in responders, and red in non-responders. And then this new paper that just came out a few weeks ago showing that favorable microbiota in the gut actually correlates or is associated with activation of monocytes and macrophages in different tumor types, including breast models, and activation of this STING pathway and interferon-dependent monocyte reprogramming. So really interesting link between the gut microbiota and the immune response within the tumor. And so just to conclude the talk, our goal, of course, is to enhance anti-tumor immunity for the treatment of breast cancer, to find novel strategies to do that. I've just hopefully presented the diversity of macrophages to you and shown the correlation of macrophages with disease progression and resistance to therapy. And I think there's great opportunity here, but I think we need to better understand these cells within the tumor. There's distinct breast cancer microbiome between benign and malignant cells. And I think greater understanding of these microbial agents within the breast tumors can expand our ability to prevent, maybe diagnose, and hopefully treat patients. And so interactions between macrophages and microbiome, I think, can hold great potential for enhancing anti-tumor immunity in breast cancer, and I think has opened up sort of some new possibilities for treatment going forward. So we'll save questions for the end, but thank you very much.